we're going to discuss the topic, What is a receptor? By Professor Swaminathan Kirahanam. Next slide, definition and meaning of receptor. Receptor, it is a macromolecule in the membrane, or inside the cell, which specifically, that means chemically, binds with ligand, known as drug. The binding of a drug to a receptor depends on the types of chemical bonds that can be established between drug and receptor. In this slide, diagram A, an unoccupied receptor does not influence intracellular processes. Diagram B, the receptor with the bound agonist drug is activated. It has altered physical and chemical properties, which leads to interaction with cellular molecules to cause a biological response. Next slide, the drug-receptor complex. Cells have many different types of receptors, each of which is specific for a particular agonist and produces a unique response. A drug is a chemical applied to a physiological system that acts on target proteins, namely, receptors, enzymes, carriers and ion channels. The formation of the drug-receptor complex leads to a biological response. This is mentioned in pictures C and D. The magnitude of the response is proportional to the number of drug-receptor complexes. Explained through the example, 1. Drug plus receptor effector produces the drug-receptor complex, which is responsible for the drug effect. 2. Drug plus receptor effector produces the drug-receptor complex, leads to an effector molecule, which responsible for the drug effect. 3. Drug plus receptor effector produces the drug-receptor complex, leads to activation of coupling molecule, and further to an effector molecule, which responsible for the drug effect. So, it concluded that the formation of the drug-receptor complex leads to a biological response. Next slide, the drug-receptor interaction. All drugs produce more than one effect on the body. The primary effect of a drug is the desired or therapeutic effect. Secondary effects are all other effects, whether desirable or undesirable, like causing harmful effects, produced by the drug. The most common way that drugs exert their action is by forming chemical bonds with certain receptors in the body. This usually occurs only if the drug and its receptor have a compatible chemical shape. Drugs with molecules that fit precisely into a given receptor elicit a comparable drug response and are known as agonists. Those that do not fit perfectly produce only a weak response or no response at all. Not all drugs that bind to specific cells cause a functional change in the cell. These drugs act as antagonists to the natural process and work by blocking a sequence of biochemical events. Some drugs may act by affecting the enzyme functions of the body. When drugs are metabolized in the liver, they produce antimetabolites. These antimetabolites interrupt or inhibit the actions of particular enzymes, thus producing the desired therapeutic effect. Next slide, drug affinity. A drug's affinity refers to the chemical forces that cause a substance to bind its receptor. It tells us how attracted a drug is to its receptors. Efficacy refers to a drug's ability to effectively activate the receptor once it has bound to it. Efficacy tells us how good a drug is at producing the desired effect. Diagram in the slide explain how strongly the drug binds to the receptor depends on the molecular complementarity of the drug and receptor. High affinity ligand binding implies that a relatively low concentration of a ligand is adequate to maximally occupy a ligand binding site and trigger a physiological response. Next slide, drug potency. Potency is a measure of drug activity expressed in terms of the amount required to produce an effect of given intensity. A highly potent drug evokes a given response at low concentrations, while a drug of lower potency evokes the same response only at higher concentrations. In this diagram graph, A is high potency, B is lower than A, C is lower than B, and graph D is the lowest potency, compared to others. So, it concluded that potency is an expression of the activity of a drug in terms of the concentration or amount of the drug required to produce a defined effect, whereas clinical efficacy judges the therapeutic effectiveness of the drug in humans. Next slide, the difference between potency and efficacy. Potency, denotes the amount of drug needed to produce a given effect. Efficacy refers to the relative ability of a drug receptor complex to produce a maximum functional response. Diagram E, drug A is more potent than drug B, but both show the same efficacy. Drug C shows lower potency and lower efficacy than drugs A and B. Diagram F, F1 shows the potency difference. It means a difference in the concentration. F2 shows the efficacy difference. It means a difference in the response. Next slide, drug-receptor interaction. It is broadly divided into agonist and antagonist. In diagram G, the agonist is the drug that binds to specific receptors and produces a drug action called agonists. This means drugs that occupy receptors and activate them is known as an agonist. An agent which activates a receptor to produce an effect similar to that of the physiological signal molecule. Using the lock and key analogy, the receptor is unlocked by a key known as agonist drug. In diagram H, the antagonist is the drugs that bind to specific receptors and block agonist drug action or cellular functions are called antagonists. An agent which prevents the action of an agonist on a receptor or the subsequent response, but does not have any effect of its own. This means drugs that occupy receptors but do not activate them. Antagonists block receptor activation by agonists. Using the lock and key analogy, the receptor remains unlocked by a key labeled antagonist drug, but the key is also blocking another key labeled agonist drug from entering the lock. Which means, an antagonist is like a key that fits nicely into the lock but doesn't have the right shape to turn the lock. When this key, known as the antagonist, is inserted in the lock, the proper key, known as the agonist, can't go into the same lock. Next slide. Types of agonists. 1. Superagonist. It is a type of agonist that is capable of producing a maximal response greater than the endogenous agonist for the target receptor. Efficacy is more than 100%. For example, goserelin is a superagonist of the gonadotropin-releasing hormone receptor. 2. Full agonists. When the receptor stimulus induced by an agonist reaches the maximal response capability of the system, tissue, then it will produce the system's maximal response and be a full agonist in that system. Efficacy is equivalent to 100. For example, full agonist opioids activate the opioid receptors in the brain fully resulting in the full opioid effect. Examples of full agonists are heroin, oxycodone, methadone, hydrocodone, morphine, opium and others. 3. Partial agonist. An agent which activates a receptor to produce a submaximal effect but antagonizes the action of a full agonist. Efficacy less than 100%. Example, the partial agonists include buprenorphine, dorphanol, and tramadol. There are mixed agonists, antagonists, which demonstrate varying activity depending on the opioid receptor but also varying on the dose. Next slide, types of agonists continue. 4. Coagonist. A coagonist works with other coagonists to produce the desired effect together. NMDA receptor activation requires the binding of both glutamate, glycine and D-serine coagonists. Calcium can also act as a coagonist at the IP3 receptor. 5. Inverse agonists. An agent which activates a receptor to produce an effect in the opposite direction to that of the agonist. An example is the cannabinoid inverse agonist rimonabant. 6. Irreversible agonist. It is a type of agonist that binds permanently to a receptor in such a manner that the receptor is permanently activated. Oxymorphone is an example of an irreversible agonist. Next slide, types of antagonists. Antagonist may be classified: 1. Physical, 2. Chemical, 3. Physiological, 4. Pharmacological. 1. Physical antagonist binds to the drug and prevents its absorption like charcoal binds to the alkaloids and prevents their absorption. 2. Chemical antagonist combines with a substance chemically as chelating agents bind with the metals. 3. Physiological antagonist produces an action opposite to a substance but by binding to the different receptors. For example, adrenaline is a physiological antagonist of histamine because adrenaline causes bronchodilation by binding to beta-2 receptors. which is opposite to bronchoconstriction caused by histamine through H1 receptors. 4. Pharmacological antagonists produce opposite actions by binding to the same receptor. For example, beta blockers. The pharmacological antagonist is further divided into competitive and non-competitive. The competition is further divided into equilibrium, known as reversible, and non-equilibrium, known as irreversible. A reversible antagonist binds non-covalently to the receptor and therefore can be washed out. An irreversible antagonist binds covalently to the receptor and cannot be displaced by either competing ligands or washing. Next slide, comparison of competitive and non-competitive antagonist. Diagram I. IT is the competitive antagonist. Competitive, equilibrium type. 1. Antagonist binds with the same receptor as the agonist. 2. Antagonist resembles chemically the agonist. 3. Parallel rightward shift of agonist dose-response curve. 4. Maximum response can be attained by increasing the dose of the agonist. 5. Intensity of response depends on the concentration of both agonist and antagonist. 6. Example, atropine, morphine, acetylcholine, naloxone. Diagram J. Non-competitive. 1. Binds to another site of the receptor. 2. Does not resemble with agonist. 3. Flattening of agonist dose-response curve. 4. Maximum is suppressed. 5. Maximum response depends only on the concentration of antagonist. 6. Example, diazepam, bicuculine. Next slide, types of receptors. 1. Ligand-gated ion channel receptor, are integral membrane proteins that contain a pore which allows the regulated flow of selected ions across the plasma membrane. Example, nicotinic cholinergic, GABA, and the 5-hydroxytryptamine 3 receptor. 2A. G. Protein-coupled receptors, adenylyl cyclase, are integral membrane proteins that are used by cells to convert extracellular signals into intracellular responses. G. T. P-bound G's alpha then binds to and stimulates adenylyl cyclase. Adenylyl cyclase is a membrane-bound enzyme that catalyzes the conversion of A, T, P to cyclic A, M, P. Example, alpha and beta adrenoceptor. 2B. G. Protein-coupled receptors, modulates enzyme activity phospholipase C, G. Protein-coupled receptor signaling pathway in which the signal is transmitted via the activation of phospholipase C and a subsequent increase in the intracellular concentration of N-acetyl trisphosphate, I, P3, and diacylglycerol, D, A, G. Example, alpha and beta adrenoceptor. 3. Nuclear receptors, or intracellular receptors, are a class of proteins responding to the extra and intracellular signals carried by steroid or thyroid hormones to regulate the expression of target genes. Example, estrogen, glucocorticoids, known as steroids, thyroid hormone T3 or vitamins D and A. 4. Receptor tyrosine kinases are the high-affinity cell surface receptors for many polypeptide growth factors, cytokines, and hormones. A subclass of tyrosine kinases that are involved in mediating cell-to-cell communication and controlling a wide range of complex biological functions, including cell growth, motility, differentiation, and metabolism. Example, insulin. 5. Nitric oxide and guanylate cyclase. Nitric oxide elicits physiological effects in cells largely by activating guanylyl cyclase-coupled receptors, leading to cyclic G, M, P accumulation. Guanylyl cyclase, also termed guanylate cyclase, catalyzes the synthesis of cyclic G, M, P from G, T, P. Example, A, N, F receptors, N, P, R1, N, P, R2 and N, P, R3, in kidney. Next slide, examples of drug targets. General target class, specific target, drug example. 1. General target, plasma membrane receptor, specific target, beta-adrenergic receptor, example drug, isoproterenol. 2. General target, corticosteroid receptor, specific target, corticosteroid receptor, example drug, prednisone. 3. General target, enzyme, specific target, cyclooxygenase, example drug, aspirin. 4. General target, ion channel, specific target, GABA receptor, example drug, barbiturates. 5. General target, transporter, specific target, serotonin transporter, example drug, fluoxetine. 6. General target, nucleic acid, specific target, alkylate chemotherapeutics, example drug, chlorambucil. 7. General target, signal transduction kinases, specific target, 1, B, C, RA, B, L. It is a mutation that is formed by the combination of two genes. Example drug, A. Imatinib, specific target, 2, M, T, O, R. It's a protein that helps control several cell functions, including cell division and survival, and binds to rapamycin and other drugs. Example drug, B-sirolimus. Next slide, examples of selected ligands, or receptors. 1 acetylcholine, 2 adenosine, 3 adrenoceptors, 4 angiotensin, 5 bomosine, 6 bradykinin, 7 calcitonin, 8 calcium sensing, 9 cannabinoid, 10 chemokine, 11 chemotactic peptide, 12 cholecystokinin, 13 gastrin, 14 corticotropin releasing factor, 15 dopamine, 16 endothelin, 17 GABA, 18 galanin, 19 glutamate, 20 glycine, 21 histamine, 22 5-H, T23 leukotriene, 24 melanocortin, 25 melatonin, 26 neuropeptide Y, 27 neurotensin, 28 neurotrophin, 29 nicotine, 30 opioid, 31 prostanoid, 32 protease activated, 33 ranodine, 34 somatostatin, 35 steroid, 36 tachykinin, 37 thyrotropin releasing hormone, 38 urotensin, 39 vanilloid, capsaicin, 40 vasoactive intestinal, 41 polypeptide, 42 vasopressin. Thank you for your attention. Further information, kindly contact profsamiacademy@gmail.com. Disclaimer: this channel does not promote or encourage any illegal activities, and all contents provided by this channel are meant for educational purposes only. Any copyrighted material on these slides is included as fair use for educational purposes only. It will be removed at the request of copyright owners.